Hello, everybody. Welcome to Miss McGuire video lectures. And today we are covering the nervous system and the nervous tissue. This is part 1, and we only will cover anatomy part. So some of the slides I'm gonna skip, and I will record the second video where I go more in detail into physiology of the nervous system. Okay, so let's go ahead and begin. So your nervous system is divided into two major divisions, central nervous system and peripheral nervous system, or CNS and PNS. CNS includes brain and spinal cord. And if you, as you see on this diagram over here, brain is located inside the cranial cavity and spinal cord located inside the vertebral cavity. PNS includes pretty much everything else. So it's accumulation of your nervous tissue outside of your brain and spinal cord that will include such organ as nerves, cranial nerves, spinal nerves, and different sensory receptors found through your whole bodies, right? As well as what we call enteric nervous system. This is the nervous tissue within your GI tract, right? So that pretty much says everything else, right? If we talk about nervous tissue, and you remember the tissues are made of cells. So what cells, involved in a nervous tissue, right? Yeah, okay, anyway. So what cells make nervous tissue? So we have neurons and what we call glial cells or neuroglia. And neurons are the major cells that will generate and conduct electrical impulses through your body. And this is how your nervous system communicates with the rest of your body by the means of chemicals, special chemicals, neurotransmitters, and electrical signals, or what we call action potential. So we have those cells, neurons, and they're gonna generate and send those electrical impulses. And then we will have supporting cells that will nourish, support your neurons, provide protection, and make a framework for the nervous system. And those are glial cells or neuroglia. Now, on this picture, you see a human brain and a little portion of it, and you can notice that some parts of the brain look darker and other parts look lighter. So that's how we differentiate between gray and white matter. Now, what is gray matter? Well, gray matter are cell bodies and dendrites, and white matter are axons. And axons are covered by fatty substance called myelin, and the myelin gives this white appearance to the axon. So let's kind of make sense of it. Okay, so look. So if I want to draw a neuron, right, I would probably draw, I'm horrible at drawing stuff, but I would draw a cell body, right? And this is where you find the nucleus and raphe and endoplasmic reticulum, right? So all these organelles, and then you would have processes. So you'll have multiple processes that we call dendrites, and we will discuss it in a second. And then you will have one process that is called axon, right? So now axon is covered by myelin, right? Not every single axon. We do have unmyelinated neurons, right, or slightly myelinated, but we are talking about kind of like general example, right? So we have cell body, we have axon covered by myelin. And then if I can draw another one, right? And again, myelin. And then I can draw another one. So you get the point, right? So I have this collection of cell bodies, right? And dendrites. And they actually would appear gray. So they have like a darker color. And then I have the whole bunch of these axons covered by myelin, and they appear white. That's how we differentiate between gray and white matter, right? So gray matter is like accumulation, the whole bunch, many cell bodies and dendrites, and white matter would be axons covered by myelin, right? Now, gray and white matter are only found, we define it as a gray and white matter only when we're talking about central nervous system, right? So for example, if you're talking about nerves, you cannot use this classification. You cannot say that nerves are made of gray or white matter, whatever, right? So gray and white matter are only, we define this as a gray matter and white matter only when we're discussing brain and spinal cord, right? That makes sense. It doesn't mean that we don't have dendrites and axons in the peripheral nervous system. We do, we just name them differently, right? And how we name them, wait a second, we will discuss it. Over here, we have nucleus, ganglion, tract, nerve, right? Now, okay, so remember we talk about this gray matter, right, and white matter. So gray matter, okay, so let me put here, gray matter, right? If we have, okay, sometimes you think like, okay, I know this stuff, how to explain it the way it makes sense. Give me a second. I will explain it to you the way I think it makes sense, and then you will let me know if it does or not. Right? So here we go. So remember what I draw for you. So we have cell bodies, right? And axons. Right? So here's my dendrite. Here's another neuron with dendrites, axons with myelin. Making sense so far? Now, in a central nervous system, this collection of cell bodies, right, we call nucleus. And this axons, myelinated axons, we call tract. Right? It's what it says. Nucleus is localized collection of neuronal cells, bodies in the CNS. And tract is a bundle of axons in the CNS, right? So if you're talking about CNS, that's gonna be our nucleus, and there's gonna be the tract. And nucleus obviously is a gray matter, right? Gray matter, and tract is white matter, right? Now, we have PNS, peripheral nervous system, and we have neurons as well. So we have cell body of neuron, axon, cell body, dendrites, then axon, right? And axons are myelinated. Now, in a peripheral nervous system, this collection of cell bodies and dendrites, now we call ganglia. Ganglion is one and ganglia is plural. And this bundle of axons myelinated axons in the peripheral nervous system we call nerve, right? And we don't use this gray and white matter for peripheral nervous system, right? So it says over here, a ganglion is a cluster of neuron cell bodies in PNS, and nerve is a bundle of axons in PNS, right? So pretty much we have the same cells, neurons, right? And neurons, if we're talking about, for example, is a motor neuron, right? Multipolar neuron. So just general neuron have a cell body and axon. So we have the whole bunch of cell bodies and the whole bunch of axons. But in a CNS, we call cell bodies nucleus and axon tracts. And in PNS, we call cell bodies ganglion and axon nerves, right? And also in the CNS, we call those nuclei gray matter and tract white matter. And we not only have tract, right? We have other terminology, right? We have association fibers, we have projection fibers. This is just pretty much like a basic stuff. Hopefully we understand now what nucleus, ganglion, tract, and nerve is, right? Nucleus and tract in CNS, ganglion and nerve in PNS. Now, do not confuse this nucleus with nucleus of an atom or nucleus of a cell, right? That's not the same. Nucleus in a nervous system, in a CNS, is a macrostructure, something that you can even see with your naked eye, and it will have lots of cell bodies of multiple neurons, right? Okay, so let me just get rid of all this writing, and right. Next, functional division of the nervous system. So, well, what are the functions? Functions is sensations, response, and integration, right? So pretty much your nervous system, receives the sensory information from outside of your body or from inside of your body. It processes this information, integrates it, compares it with your previous memories, with your experiences, and then make decision what to do with the sensory information. So your central nervous system gonna send impulses to your muscles or to your glands, and they will respond based on the type of sensory information that they received, right? So the simple example would be, I can receive visual sensory information, somebody entering my room, right? That's my sensory information received by my my special sensory organs, with my eyes, with the retina. Now this information sent to my brain, and my brain making sense of it. Who is this person? Do I know him or her? You know, is it the person who might be a dangerous, or is it my friend, right? And based on this processing and integration and kind of like comparing what I see now with what I already know, my central nervous system gonna send signals to my muscles, for example, and I can say, hey, hello, right? Muscles of my arm and muscles of my tongue, so I can talk and I can wave. So that will be my motor response to the sensory information that I received, right? But your central nervous system receives lots of information from your internal organ, like from your heart, from your blood vessels, from your skin, and it's also processes, integrates it, and provide signal to your effector organs that will respond to that information, right? Anyways, that's a basic functions. Now, nervous system can be divided into somatic and autonomic. And over here, part of autonomic nervous system is enteric. just give me a second, we'll talk about it. So somatic nervous system will include sensory division and motor division, right? And it will be conscious perception of sensory information, right? So when we're talking about somatic nervous system, that means that sensory information that you're receiving will reach your consciousness. So you will understand that information, conscious perception, and then you will produce voluntary response, right? So that's the example I gave you. I see somebody, it reaches my consciousness, and then I voluntarily respond, right? Like I'm saying hi or wave my hand. Autonomic nervous system will receive sensory information as well, but it's gonna be visceral sensation. So it will come from your internal organs, and it will not reach your consciousness. An autonomic nervous system will produce involuntary response, something that you do not control. For example, if your nervous system receives information about your body temperature, your temperature increasing, right? You might not even understand your temperature goes up, really. But then your autonomic nervous system gonna send signal to your sweat glands, and you will, produce sweat, and it will cool you down, right? That's a part of autonomic nervous system. Enteric nervous system are found in your digestive system. And if you read over here, enteric nervous system located in the digestive tract is responsible for autonomous functions and can operate independently of the brain and spinal cord. And that's the system that will produce a contraction of your stomach and small intestine and large intestine, and also secretion of digestive enzymes, right? So, and that will be done automatically, and not only automatically without your control, it can be done even without your brain and spinal cord control. So your central nervous system doesn't need to control it, right? So again, we can divide nervous system into somatic and autonomic, and part of the autonomic is enteric. Now let's look at structure of a typical neuron. So I already mentioned the neuron has a cell body, and cell body contains most of the organelles, like a nucleus, like ribosomes, then mitochondria, right, rough ER, smooth ER, right? So that would be found inside the cell body or soma. Then the soma will have processes, and some processes, multiple processes. In this example would be dendrites, and then it would be axon, and human neurons, they have only one axon. Now dendrites will receive signals and send those signals towards the cell body, and axons will send signals away from cell body toward the axon terminal, right? Towards the end of this axon, where we will have like a synapse or connection, not physical connection, it will be a gap, but this is where one axon communicates with another axon, or maybe with your muscle cell, like with some effector cells, right? So dendrite sends information towards the cell body, right? Here we go, like this, and axon sends information towards synapse, right? Also, axon hillock is the where axon emerges from the cell body, so that pretty much like this first part, right? So it's really part of cell body yet, but it's the most excitable part of the axon, right? That's called axon hillock. Now in axon, we have what we call exoplasm, because it's not the same cytoplasm that is found in a cell body. Cytoplasm changes to contain limited components, right? Like it doesn't have nucleus, it doesn't have some of the organelles, right, that found inside the soma. Now, axons can be myelinated. Not every single axon is myelinated, right? But most of them are. So we have this myelin sheath, right, shown over here along axon. And this myelin sheath is produced by neuroglia. In this example, it's oligodendrocyte, right? That's a special cell found in the central nervous system that secretes this rich organic substance that mostly made of fat and protein, right? Myelin. And myelin sheath, here's the name, by the way, myelin sheath, it's not continuous covering. There are gaps in between, and those gaps are called node of Ranvier, all right? And they are critical for signal transduction. And then we have axon terminal, right? Something, the end, like the end of the axon. And we have these synaptic bulbs, right? So that's the bulbs over here. And where we have this axon terminal, this is where one axon will communicate with another cells. It can be another axon. Oh, I'm sorry. Yeah, it can be another axon. It can be another neuron. That's what I wanted to say. Usually it's a dendrite of another neuron or soma of another neuron, or it can be axon of another neuron, right? But the point is, at the synapse, one neuron communicates with another neuron or with another cell, right? Okay. Now, types of neurons. Now we have unipolar neurons, bipolar neurons, and multipolar neurons. In human, we don't really have unipolar neurons. We even call them now pseudo-unipolar. That's why it says found in invertebrates only, because we are vertebrates. Right, but because we have pseudo-unipolar, it's better to know what it means, right? So, and we do have this pseudo-unipolar neurons, right? That's, I mean, sensory neurons are unipolar. So let's call them unipolar, right? I don't know why wouldn't we be able to call them unipolar? There should be a reason for that. We will do research about that reason. So anyway, we have unipolar. What it means? It means when we have a cell body here, right? We actually have like one process from cell body, and then it splits into peripheral process and central process. We don't even call it dendrite and axon, but it's kind of like it. So it's kind of like dendrite. So peripheral process will bring information towards the cell body, and it would be like your dendrite. And central process gonna send information towards your central nervous system, and it's gonna be like axon, right? But the point is, we have one process only that emerges from the cell body, right? And in human, sensory neurons are unipolar, right? Sensory neurons are unipolar. Now, bipolar neurons are pretty rare, and we have two processes from cell bodies, right? That's why it's called bipolar, right? And one gonna be axon. I'm sorry, which one it will be? Yeah, this is gonna be dendrite, and this is gonna be axon. And those, as I said, very rare example would be olfactory neurons or neurons found in your retina. Those are bipolar neurons. And most of neurons are multipolar. So all neurons that are not unipolar or bipolar are multipolar. And we see this cell body, and we have multiple processes, one axon and many dendrites, right? That's a multipolar neuron. So your inner neurons, your motor neurons, they are multipolar neuron. So neurons, this is structural classification based on structure. We can also classify, neurons based on who discovered them, or based on their location or function group, right? Or by other shape rather than multipolar. So like this is multipolar neuron A, right? This is multipolar neuron B, but they have their special name, like pyramidal cells, based on their shape found in the cerebral cortex, or Purkinje cells found in the cerebellar cortex. So this is cerebrum, this is cerebellum, and this is named based on the person who discovered them. Or we can call them olfactory neurons, like this one right here, right? Those are bipolar neurons, by the way. All right, so here it goes all the way right over there. So that's a bipolar neurons, but we call it olfactory based on their location. Now, glial cells, those are supporting cells, right, that nourish neuron, protect neurons, provide this framework for nervous system. Many of them involved in extremely important functions, all of them involved in important functions, right? They just cannot really do this major work, right? That neurons do, like generation of action potential, sending signals to your brain, away from your brain. But obviously your nervous system cannot function without them. And those glial cells or neuroglia in the central and peripheral nervous system are different. So actually, let's look at next slide. In the peripheral nervous system, you have glial cells, both start with S, right? So you see SS, then it's gonna be PNS, SS, PNS. Everything else is CNS. So what we have in CNS, we have astrocyte, we have oligodendrocytes, microglia, and ependymal cells. Astrocytes have a star shape, and they're important in the formation of blood-brain barrier that we will talk about later. They also provide support to neurons. So on this diagram, cartoon diagram, here's the astrocytes, and you see how it has connection to a neuron and other cells, kind of keeps everything in place, right? And also make sure that pathogens, or probably viruses or whatever in your blood would not reach your brain tissue. That's what blood-brain barrier is. Oligodendrocytes shown over here, right? So here's oligodendrocytes, those cells that secrete myelin and insulate axons. They also very critical because they, speed up this conduction, the velocity of the conduction, right? So you can see one oligodendrocyte can actually form myelin sheath on a multiple axons. Microglial cells, like shown over here, those are phagocytic cells, and it's part of the immune system of your brain and spinal cord, right? So they can engulf pathogens, bacteria, or viruses, or some cellular debris. And ependymal cells found over here, they involved in the production of cerebrospinal fluid. Cerebrospinal fluid is made from your plasma, and then your plasma moving through the cells, of course, will change its chemical composition, right? And it will be released in a subarachnoid space, right? So it will be released in a space between your, brain tissue and other tissue. So it will cover your brain, it will cover your spinal cord, right? And ependymal cells, they are major players in the production of CSF, cerebrospinal fluid. Right, this is how it might be silly, but this is how I remember it. Microglial cells, they are small, micro, but have a big job, protection, phagocytic, right? So immunity. And right, so that's how I remember at least microglial. And, you know, I don't know, astrocytes, like a star shape. That's a good shape to give a support, right? Anyway, whatever helps you to memorize function of the cells will work. Now, in the peripheral nervous system, we already mentioned we have two cells, both start with S, so it's a satellite cells and Schwann cell. Now, satellite cells similar to astrocytes, and astrocytes mean a star, right? And satellite, for me, means moon, right? Because moon is our satellite. So I'm thinking stars and moons, right? Satellite, astrocyte, you know, something that might have similar function, right? So that's how I remembered it when I was a student. So satellite cells support similar to astrocytes. The difference is satellite cells are found in the PNS. And here's the satellite cells shown over here, nice, right? And that's a cell body, right? So here's a cell body of neuron, and they covered by the satellite cells. And Schwann cells, they have similar job to oligodendrocytes because they secrete this myelin for axon insulation. And here's I show you a Schwann cell. So that's one Schwann cell. This is another Schwann cell. The difference is Schwann cells form myelin only on one specific axon, right? When oligodendrocytes, you see, can, like this cell can form myelin on this axon and this axon that belong to a different neuron. Schwann cells cannot do it. They just wrap themselves around axon, and they don't have these processes to deposit myelin to another axon. Now, myelin sheath is critical in the conduction of these electrical signals. It's made by glial cells, oligodendrocytes, or Schwann cells. We already discussed it. Now, this one thing over here that I think I need to mention. So you see this yellow structure is axon, right? So it shows here it's an axon. And what cover axon, the membrane, is called axolemma. So here's the axolemma. So axolemma is inside, it covers, so this, like shown here in black, right? So that's an axolemma. And then we have a Schwann cell. So this blue structure is a Schwann cell. It has this nucleus. And Schwann cell kind of wraps itself around axon several times, producing this myelin, right? And the outer part is called neuralemma, right? And neuralemma will contain a nucleus of Schwann cell and most of the cytoplasm, right? So we have axolemma covering axon, and we have neuralemma, the more superficial covering of myelin sheath. Right? And demyelination means a damage of myelin sheath, and this will slow electrical signals. And we have several diseases that are caused by demyelination. You probably heard about it, multiple sclerosis. This is demyelination in central nervous system, and Guillain-Barré syndrome. This is demyelination in peripheral nervous system. So the CNS and PNS here. Okay, now, as I said, we're gonna skip some of these slides because this video is about anatomy only, right? So over here, we're talking about propagation of action potential. So just very quickly, because we're not going into detail. So here's a neuron over here, right? A neuron, at this point where you have axon hillock, gonna generate electrical signal. And this electrical signal will go from along the axon. What is it? Okay, along the axon, and probably, here it's towards another neuron, right? So this electrical impulse is called AP, or action potential. So it's right here. So action potential is this electrical signal that moves along axon, from one axon to another axon, or from one axon to another cell, right? And this is called AP propagation. So it's propagates, so it's moving, right, along the axon. So if we have nice myelin covering right here, right, we have one Schwann cell producing this myelin sheath, another Schwann cell, and there are gaps in between them, right? We already talked about it, node of Ranvier. So if we have these nerve impulses pretty much going from one nerve to another nerve, they call it jumping of action potential. Then this is called saltatory conduction. So saltatory conduction, action potential jumps. It doesn't really jumps. It just the word that used to describe this action, right? So it jumps from one node of Ranvier to another node of Ranvier, and the conduction is fast. So that's really good. Myelinated axon conduction is fast. So we go from one node to another node, so we get to destination faster. If we do not have myelin sheath, and some axons just don't have it, right? And or myelin sheath can be damaged, like in this example, like in a multiple sclerosis, for example, or in Guillain-Barré syndrome. So we have damage of this myelin sheath, then we have continuous conduction, right? So it's easily found in unmyelinated axons, and it's a slow conduction, right? Or it became continuous instead of saltatory when we have damage of myelin sheath. Right. And of course, it says over here, when we have saltatory conduction, then axon just moves faster. I'm not going into detail what depolarization is, because obviously we didn't discuss it yet, right, in the second video. So here you can see the difference between saltatory and continuous conduction. So saltatory conduction is propagation of action potential between the nodes of Ranvier in myelinated axons, and continuous conduction is propagation of action potential, that is this electrical impulse that we just mentioned, right, along the entire length of unmyelinated axons. And this goes between the node of Ranvier, this along the entire axon. Saltatory conduction is faster, continuous is slow. Then it's more efficient, less efficient continuous conduction, right? And energy use, right, is low in saltatory and high in the continuous, right? So that's pretty much the difference between saltatory and continuous conduction. And some axons that I already mentioned several times, they just not myelinated, and they don't need this fast propagation of action potential, but sometimes just myelin sheath became damaged, and then it will affect the health, and it will cause some condition, disease, right? So that's, by the way, this condition, multiple sclerosis, we're going to discuss very quickly. So it's autoimmune disease that mainly affects young adults. That means your own immune system attacked this myelin sheath, and you can see that myelin sheath now get damaged, and then the speed of electrical impulses decreases. So, and because, well, multiple sclerosis, again, it's found in a central nervous system, right? So that's a disease of central nervous system. And because your central nervous system pretty much responsible for so many functions, like your vision, your muscle control, your speech, your urination, defecation, memory, like everything, right? So that depends where exactly you have this damage. You might have, well, somebody might have different symptoms like visual disturbances, weakness, loss of muscular control, speech disturbance, urinary incontinence. All right. And but the underlying cause is the same. Myelin sheath in the CNS become non-functional sclerosis, sclerotic, and it's cause shunting and short circling of nerve impulses, and impulse conduction slows and eventually ceases, right? So, and if your nervous system cannot send or receive these electrical signals, that will affect many important functions, right? Like your vision, like your skeletal muscles, like your smooth muscles in your urinary bladder, right? Okay. So, and kind of couple more slides we need to cover over here. Synapse. So when we have one neuron, right, and we have axon, and then let's say this neuron, so here's my dendrites, communicate with another neuron, for example, right? There is no direct connection between axon and dendrites or cell bodies. There is a gap in between, and this gap is called synapse. So you can see this is, for example, axon of one neuron, and this is maybe a dendrite of another, and there is a gap in between. Or we can have a gap over here that actually connected by special channels, right, over here. But still, there is a gap in between. Now, depends how we transmit signals within this gap. So what's going on, let's say, depends on what's going on inside this gap. We have either chemical synapse or electrical synapse. Chemical synapses are most common. They're very common. Most synapses are chemical synapses. And why we call them chemical synapses? Because chemicals are released in this gap. And that chemical is called neurotransmitter. So when signals reaches the axon terminal, then synaptic vesicles will fuse with the membrane of presynaptic neurons, this one called presynaptic, so that's a presynaptic. A neurotransmitter is released inside the synaptic gap, and then neurotransmitter attaches to its receptors. So those blue proteins over here are receptors. So you can see neurotransmitter attaches, and it will cause changes within postsynaptic neuron. So like this neuron sending information in the form of electrical impulse, but when it comes to the end, to the axon terminal, this electrical signals became chemical signal of neurotransmitter. And this neurotransmitter now will cause change in the postsynaptic terminal or neuron, right? That's a chemical synapse. And electrical synapse, when we have gap junction between one and another, and electrical impulse just goes straight from one to another. This is found in your heart, in embryonic tissue, but it's not very common synapse in the adult humans, right? Rather than you find it in between the cells of your myocytes of your heart muscle. Right, let's look at a chemical synapse, right? So we already did, but let's look again. So we have synaptic cleft, right? This gap in between synaptic cleft, and this is where neurotransmitter is released. And now this is our axon terminal. And this is called presynaptic neuron. So it's before synapse, presynaptic neuron, and this one is called postsynaptic neuron. So you can say like presynaptic neuron gonna release neurotransmitter and postsynaptic neuron gonna receive this neurotransmitter. Now, neurotransmitters can be different, but the point is they all will have their receptors. They need to attach to the receptor and cause some changes within postsynaptic neuron, right? Okay, so that's a chemical synapse. And in the second video on physiology, we will go into more details, how it works. And here's some neurotransmitters. Some of them, I think more than 50 neurotransmitters are discovered so far, and we're still in the process, but we can classify them based on their chemical structure. So we have cholinergic receptors, and example would be acetylcholine. And this is the one that is released. It's very common neurotransmitter, but for our purpose, let's remember that acetylcholine is released in the neuromuscular junction. So where your skeletal muscles are, those neurons release acetylcholine to activate your skeletal muscles. Now, amino acids, right over here, an example would be glutamate or GABA. GABA is inhibitory neurotransmitter, and glutamate is excitatory. But again, we classify them based on the structure. Biogenic amines. So amino acids, they have this carboxyl group, COOH, and biogenic amines, they lacking of this carboxyl group. An example gonna be epinephrine, norepinephrine, dopamine, serotonin. Oh my goodness, all this stuff that we know, right, affect not only our, electrical activities of neurons and muscles, and it also affects your mood, your memory, your emotions. And neuropeptides, well, peptides means it's a small proteins, right? So they're made of amino acid. And example over here, beta endorphin or substance P. Substance P allow you to feel pain, right? So, but again, we classify them based on their structure. They can be either excitatory or inhibitory, depends on their receptors, right? They do have different receptors over here. And the interesting stuff over here, we need to remember, we always need to eliminate this neurotransmitter from the synaptic cleft. So we don't want this neurotransmitter to stay here forever. Otherwise, you constantly will excite your postsynaptic neuron or other cell, whatever you're exciting, your muscle, for example. So you need a mechanism to remove neurotransmitter from the cleft, and there is different type of eliminations that's shown over here. Right. Okay. And for our first video, this is the last slide that we covered. Right, so again, this was a nervous system and nervous tissue part 1, anatomy only. Thank you for watching, and I hope it was helpful.